Hello class, this is lecture 2. This week we'll be discussing gel electrophoresis and DNA sequencing. So here's a closer look at what we'll be discussing. For gel electrophoresis, we'll be talking about the basic principles, then we will discuss DNA sequencing, and last we'll go over pulsed field gel electrophoresis, or PFGE. For sequencing, we're going to talk about the Sanger or chain termination sequencing method. We'll discuss contigs and next generation sequencing, and finally single molecule sequencing. So first let's take a look at the basic principles of gel electrophoresis. So I wanted to revisit two different points from lecture 0 that will help us understand gel electrophoresis a little bit better. The first is that the DNA backbone is made up of sugar and phosphate groups, and you see these here on the diagram circled in red. Because of the electrons on the oxygens, each of these phosphate groups has a negative charge. This has the net effect of giving a molecule of DNA an overall negative charge. The second point is that the configuration of DNA molecules can vary greatly, from large chromosomes to plasmids, and then there are supercoiling, and all of that's going to play into gel electrophoresis, as we'll see coming up in the upcoming slides. There are a number of chemicals from which you can make gels for gel electrophoresis. One of the most commonly used is called agarose. Agarose is a sugar. It forms long chains of these disaccharide units. You can see in the diagram within the brackets, two of these sugar rings are bound together. These two rings in this formation form a unit of agarose, and so an agarose chain is made up of repeating units of these two particular sugars. So you see just after the brackets to the right, another ring that is the first ring of the next unit of agarose, and then previous to the brackets you have what within the brackets is the second ring, and so that's the final ring of the previous unit of agarose. Now agarose together does start to eventually branch off and form these branched chains, and so it ends up not being just a single linear molecule. You have these branches, and so it'll fork and start to form this sort of net, particularly when it cools. So you have at higher temperatures longer strings of agarose, and then as the temperature starts to drop, you start to get this branching, and the branches start to intertwine, and then as the temperature drops even further, that intertwining starts to make a large mesh of chains with gaps in between, and this forms the basis of what we use for gel electrophoresis. You can see here in this slide a scanning electron micrograph of a solidified agarose gel, and so you see all the strands of agarose intertwined into this large mesh, and you can see all the pores in between all the different strands, and so you can imagine a molecule trying to worm its way through these various pores would encounter some resistance, but should be able to move through. So now that we understand the structures that agarose can form and what an agarose gel looks like at the microscopic level, let's talk about how we can use that for molecular biology. You can melt agarose. Typically that's done in a microwave, and so you pour your sugar powder into water, kind of stir it up some, put it in the microwave, and boil the water, and that helps the agarose get into solution. Then what you do is you pour liquid agarose into a mold that's in the shape of what eventually your gel will be, and allow it to start solidifying. And you can see here in this diagram what a gel would look like. Where it's labeled wells for samples, typically what we do is we put what's called a comb, which has these teeth. I mean, it really does look kind of like a comb, and because those teeth kind of come down into the liquid will form around it, and when it solidifies, you can pull that comb out, and that negative space will remain there, and we call those wells, and that's where we put our samples for electrophoresis. So once it cools, it solidifies, and then what we do is we put that into an electrophoresis chamber, which has buffer solution that's used to conduct the electricity from the electrodes on either end of the gel. And that current running through the gel is what helps to push the DNA down the gel. And let's take a look. Remember the opposites attract rule. So because DNA is negatively charged, it will be repelled by the negative electrode, and it will be attracted to the positive electrode. And so you see in the top part of the diagram our DNA in the well, and the anode just above that to the left, and on the other side of the gel you have the positive electrode, the cathode, and when we run current through there, the negatively charged DNA will be pushed away from the anode and pulled toward the cathode, and because of this, the DNA will migrate through the gel, working its way from the anode to the cathode, and because of the way that the gel separates the DNA by size, we end up with different sized fragments, and we'll take a look at that. So as I just mentioned, when your DNA sample contains various different lengths of DNA, whether that is from a restriction digest or a multiplex PCR, anything like that, when you put these into the well of the gel and run current through the gel, all of the DNA will begin to move down through the gel from negative to positive, but they won't all move at the same rate. Instead, they move through the gel in a size-dependent manner, and the rate at which the molecules will migrate through the gel is inversely proportional to their length. And so you can see in the diagram that these smaller molecules move through the gel more quickly, whereas the larger ones stay further up at the top of the gel because they can't move through as quickly. And once we treat the gel with a fluorescent dye, we can see bands that correspond to a specific size in the gel, and we'll discuss fluorescent dyes coming up next. The other factor, apart from length, that greatly affects the migration rate through a gel is the supercoiling of DNA. And you can see in the figure at the bottom a plasmid in various different stages of supercoiling. So the image on the right would be the plasmid in its most supercoiled formation, whereas the image on the left would be what we refer to as the open circular formation. And so the important thing to realize with respect to gel electrophoresis is that supercoiled DNA will migrate much more quickly through a gel than will the open circular DNA. And so here we're just talking about the particular structure that it forms three-dimensionally. We're talking about the same plasmid, and so the length is not any different. It's just more spread out. And you can see on the gel image on the right, this is a gel of a plasmid, which is 6.4 kb in length. And you can see the open circular DNA is up at the top, whereas the supercoiled DNA is much further down the gel. Now, the linearized plasmid also runs slightly faster than does the open circular DNA. Linear plasmid is a plasmid that has been linearized, and so if you cut it just once, such as with the restriction enzyme, now you have, a circle cut once, so you have just one long string of DNA that in this case is 6.4 kb in length. And because that's linearized, it does tend to run a little bit faster than the analogous open circular DNA. And so now let's take a look at some of the mechanics. Here's a diagram meant to represent a gel. You can see the electrodes at either end, the anode at the left and the cathode at the right. The gray circles are meant to represent the individual strands of agarose that are all intertwined, forming the gel, and the spaces in between represent the pores through all those agarose strands. So let's take a look at the migration rates of the three configurations of DNA that we talked about. We see our open circular, our linearized, and our supercoiled. So the open circular, because of its wide spread out configuration, meets a lot of resistance, and it's bumping up against more of these strands than some of the other formations. And so because of all that resistance, it has a much harder time winding its way through these pores, and doesn't migrate as quickly as some of the other formations. The linearized molecule still meets with a lot of resistance because it is also spread out, but because it is linearized, it has that one cut, which means it does have an end that's sort of able to snake its way through the various pores, and so it's sort of the middle ground between the open circular and supercoiled, in that it is still meeting a lot of resistance, but is able to move more quickly through. The supercoiled, on the other hand, really comparatively has very little resistance because it is so tightly wound that it can just kind of snake through those pores really easily. And so for an identically sized molecule, it actually does make a big difference if the molecule is supercoiled in terms of the rate of migration. Okay, so we talked about making DNA migrate through a gel in a size-dependent manner, but you can't see the DNA bands that form without some help, and so for that we're going to discuss DNA staining. Typically we use chemicals that bind to DNA and fluoresce under ultraviolet light. Some examples of these would be ethidium bromide, cybergreen, and cybersafe. The one that is most commonly used in laboratories is ethidium bromide. So now let's take a closer look at ethidium bromide. It does contain ethidium, which is a planar cation, and you can see the structure of ethidium in the molecular diagram on the top right. As I said, it's a cation, so it does have a positive charge, in this case on the nitrogen, and it is planar, so the three rings that are bound together up at the top of the molecule are in a single plane, and you can see that also in the diagram a little bit more clearly at the bottom right. The atoms that are in dark orange are the rings of the ethidium, and you can see they all lie in the same plane, forming this flat part of the molecule. Ethidium does bind to DNA via what's called intercalation, and intercalation simply means that it actually manages to squeeze itself in between the two different strands of DNA, and furthermore in between the adjacent base pairs. So you can see that in the molecule diagram here, and we can look at that even more simply. So here's the 5' to 3' directionality of one strand, and you can see that the nitrogenous bases are also in this planar fashion or orientation. And so here's the second base in essentially the same plane. The opposite strand is of course in the opposite 5' to 3' directionality, and its nitrogenous bases are also in essentially the same plane. And ethidium, the ethidium ion, manages to actually squeeze itself into the space between both the bases and the DNA backbone. To do the actual staining, we take a gel that has been electrophoresed, and so its DNA has already been separated into bands based on the various sizes. We simply can't see those bands yet under visible light. So then we soak the gel in a solution of ethidium bromide. We allow sufficient time for the ethidium to intercalate in between the strands of the DNA. Then we remove the gel from the ethidium bromide solution. At that point we still can't see the bands, but once we apply ultraviolet light to the gel, then the bands begin to fluoresce, and we can capture those images using a camera. And using this transilluminator system, you can see a gel here in the picture that has been stained with ethidium bromide, and all the various bands are visible. And based on your experimental needs, you'd be able to estimate the size of these bands according to. So finally, I want to discuss a variant of electrophoresis called pulsed field electrophoresis, or PFGE. So before we talk about how PFGE works, let's talk about why it's necessary. We mentioned the size-dependent migration of DNA molecules. Here on the right, you have a diagram meant to represent a gel. And so you have the well up at the top of the gel. We load our sample into the well. Once we apply our electrical current, the DNA molecules will migrate through the gel, forming these bands. But the problem is that that is only true up to a certain length of DNA. As you can see up at the top, we've got this big messy cluster of DNA where the various molecular weights were unable to separate sufficiently to resolve them as separate bands, and so you end up with sort of this blob at the top of your gels. So PFGE basically arose from the question, how can we separate those out so that we can actually visualize the various fragments. So PFGE works by migrating the DNA in multiple directions. This is accomplished by changing the direction of the current, and so the direction of the current alternates between different orientations. However, the net movement of the DNA down the gel remains in one direction. So here we have a diagram meant to represent a typical PFGE chamber. These do tend to be much larger than normal electrophoresis chambers. Um, this is probably about a foot and a half or so around, and so the gels tend to be much larger, as you would expect for a gel necessary to separate out large chromosomal fragments. And so we have in this diagram a single well, because we'll just be looking at one for simplicity. And so you see the hexagonal shape. There are 3 different anodes, whereas with the electrophoresis process we talked about previously, we're only looking at one anode. And similarly, we have 3 cathodes down at the bottom. And so when we first start our PFGE process, we use the opposite side electrodes, and so we have our negative charge up at the top and our positive charge at the bottom, and what that causes is the direction will turn off those electrodes, will turn on 2 of the other electrodes, and what this results in is the DNA movement direction to be from the negative to positive of those electrodes, but because the gel stays oriented in the same position, it actually causes the DNA bands to move in that direction towards that positive electrode. And then after a few minutes of movement in that direction, we will turn off those electrodes, turn on the opposite ones. As you would expect, the DNA movement is now in that direction, and so then the DNA molecules will move back in that direction, and as you can see, they've now lined up again, essentially straight down the gel, and the critical part of this is that the 2 steps the direction of DNA movement is off to the sides have to be for the same amount of time. So say 60 seconds, to the left, and then 60 seconds to the right, in order to end up in the same line, lined up with the well. And then we can cycle back to the first electrodes perpendicular to the gel, and run that out a little bit further, and then from there we would keep switching it back and forth, and so now you can see up at the top you have alright, so now we're going to move on to DNA sequencing. We're going to start off talking about Sanger sequencing. So recall that in both lecture 0 and lecture 1 I discussed the formation of the phosphodiester bond. The reason that I wanted to really emphasize that was specifically for what we're going to discuss now, is I really wanted you to understand the process of chain termination sequencing and where it comes from. So let's just review that really briefly. Remember that a covalent link is formed between the nucleotides using the phosphodiester bond. and that requires the presence of the OH group on the 3' carbon, and so you see that here. So in the strand synthesis, the next nucleotide would come along, hydrogen bond to the base opposite the new base. The electrons from that oxygen would attack the phosphate. That would separate out inorganic phosphate and form this phosphodiester bond. And then the OH at the bottom of this newest base would form the phosphodiester bond with the next base. So in Sanger sequencing, we add our deoxyribonucleotides, our DNTPs, which, as we discussed, have an H at the 2' carbon and an OH at the 3' carbon. But mixed in with those DNTPs, we also have dideoxyribonucleotides, or DDNTPs. The difference between these is that they have an H at the 3' carbon instead of an OH. And recall that we said that that OH is critical for forming the phosphodiester bond with the next base. So if we have our DNA strand with a D-DNTP at the end of the strand, there's now an H at that 3' carbon. And so when the next base comes along, there is no way to form the phosphodiester bond between that base and the previous base. And so that inability means that the chain gets terminated at that particular base. So that is now the last one, and there can be no further nucleotides past that. Hence the term chain termination. So as I mentioned, for Sanger sequencing, your reaction mixture will contain both regular DNTPs and DDNTPs. Typically the DDNTPs are added at a much lower concentration, about 10%, to the regular NTPs. And so for each of these particular types, the DDA, DDT, DDC, and DDG, where those incorporate is fairly random. And so you can see in the diagram here the T's on the template strand. Obviously the A's would be binding on the newly synthesized strand, and so you can see on the top one it binds to the it binds to the third T and ends up terminating the chain there. On the second one, it's actually binding up above all three T's, and then on the third one in the middle T. And so with a long enough sequence, you can end up randomly terminating at essentially all the different, in this case, A's of that particular sequence. And so you can see on the diagram on the right how they all end up having different lengths, and again, that because it's chain termination, there is no extension beyond that. Now the important part of this also is that each of these DDNTPs is labeled with a different fluorescent dye, and let's take a look at how that helps. So here we see some of the different DNA molecules of various lengths that have been terminated at various different positions. And so up at the top one, we have a C at the end, and so we have a DDC at the end of that chain. The next one down ended at the T after that C, and so we had a DDT incorporate there and not allow any extension past that chain termination point. And then an A, G, A, C, A, T, G. And so because these each have a different color fluorescent dye, we can then run these through capillary gel, which is a very, very thin gel inside of a little bitty capillary. Um, and the process is essentially the same as the gel electrophoresis that we talked about. And because each of these DNA strands that we see on the left are a different length, even if it is only by one base pair, because of the minuscule gel inside of the capillary, it is actually able to resolve single base pair differences in length. And so even though it's only one base pair, they will separate out into different bands. Now, because the each of those bands of that same length are terminated at the same point or the same letter, for example, A, all of those A's would have a red dye, and we can run those through a laser that can read that fluorescent dye, and then when we send it to our computer for analysis, we can generate what's called a chromatograph, which is the height of signal from each of those bands based on the color. And so you see in the chromatograph reading down the readout C, T, A, G, A, C, A, T, G, which corresponds to the lengths of or the different DNA molecules over on the left that were terminated at different parts of the chain. So here's a more realistic view of what a chromatograph would typically look like. You can see all the different peaks and the corresponding nucleotide that has been called up at the top. And so the software will examine the heights of the peaks at that particular nucleotide and make the call as to what nucleotide is at that position. So let's take a look at one example. You can see here we have a nucleotide that has been called a T, and so if we take a closer look at that, you can see here at this spot, but also all throughout the rest of the sequence, that you do have generally all four peaks represented for all four nucleotides. It is pretty difficult to get a completely clean signal. You essentially always have all four nucleotides read at that position. That's just some of the physical limitations as far as the resolution, but the difference in the peak heights is what enables the software to make the call as to what nucleotide is at that position. So you see here obviously the red fluorescent dye corresponding to thymine is by far the highest. You do have some much lower levels of the other three dyes. The green corresponding to A, the blue corresponding to C, and the black to G. And so based on the fact that the A, C, and G are so low and the T is much, much higher, you do have the software be able to determine with high confidence that the nucleotide at this position is a T. Now, if we look at sequence at the very beginning of this particular region that was sequenced, we actually have a lot of ends where the software was not able to make an appropriate call. If we take a closer look at these chromatograph peaks, you can see that it's frankly a mess. Particularly right there in the middle, you have a very high red peak and a very high green peak, but even the blue and the black are certainly much higher than you would like for a clean read, and so the software is not able to determine with any confidence what nucleotide is at that position, and so it calls it an N, which basically means any of the 4 nucleotides. Um, this is a very common problem at the beginning of a sequenced region for reasons that we won't get into. Um, but it's essentially an unavoidable problem at the beginning of your sequencing reaction. So the way that this is typically um, worked around is that the sequencing will happen in 2 different directions, and then contigs will be formed, and we'll get into a little bit more of that later. So it's now later in the lecture, so let's talk about contigs. The term contig comes from the word contiguous, and so a contig is a larger sequence of DNA that is created by overlapping and combining multiple shorter sequences. So let's take a look at an example of that. Here we have one sequence that we have generated, and here we have a second sequence that we've generated, and as you can see, at the end of sequence 1 and the beginning of sequence 2, we have this overlap region, and so we can combine these sequences and generate a single contig, or single sequence of contiguous DNA that represents the overall sequence. You are required to have an overlapping region in the middle to be able to do this, and generally the reason that this is done is because some of the physical limitations in the sequencing reaction make it hard to sequence longer stretches of DNA. Typically about 700 base pairs for Sanger sequencing is kind of the limit. So if you wanted to sequence something that was, say, twice that size, say about 1500 kb, then you would have to break that up into at least 2 reactions that overlap each other. And so you would generate, for example, the way we have here in this diagram, sequence 1, sequence 2 that includes part of sequence 1, and if your region was long enough, you could do a sequence 3 that overlaps part of sequence 2, and then combine your results in this fashion. So let's take a look at a real-world example of that. We've discussed that bacteria have the 16S ribosomal RNA. That particular sequence can be very differential between bacterial species and even strains sometimes. And so sequencing that particular region of the genome can enable you to identify the species of a sample. And so it's, at least in bacteria, one of the most commonly sequenced parts of the genome overall. And but the problem, as I was mentioning on the previous slide, is that it's about 1500 base pairs, and so you can't do that in a single reaction, and so that is broken up into 2 reactions. So let's say we have our 16S rRNA sequence here. There are 2 primers that are called D27F and 770R. The numbers come from where exactly they bind within the sequence. It's about 1500 kb. I'm sorry, excuse me, 1500 base pairs. And so the forward or F primer binds at about 27, and the reverse or R primer binds at about 770. That generates a product which you can then sequence. Then there are 2 other primers, the 704F and the 1492R, and again the numbers come from where they bind. Those generate another sequence. You have this overlapping region, and you can combine the 2 sequences that you get and generate the full-length 16S rRNA. And the actual letters you see there are part of the sequence, but that's clearly not 1500 base pairs, but that's just there. as an example. And so, as I said, this is in bacteria. Uh, the principle is exactly the same for any gene. It doesn't matter what organism, whether it's prokaryotic, eukaryotic, anything. If it's longer than your ability to sequence, then you have to break it up just like this. But this is a particular example that I'm fairly familiar with. But as I mentioned, having the overlapping regions in your sequences is critical to forming a contig. It doesn't matter how many different reads you have that you're trying to combine into a contig. They all do have to at least overlap each other once. So if you have two different sequences you're trying to contig, they each have to overlap each other. If you have three, you have to have an overlap between 1 and 2, and then also between 2 and 3, and so on. And so the problem that you run into is if you don't have an overlap, they simply can't be contigged. So, for example, if we have this gene here, we use these primers to generate this sequence, and then we use these primers to generate this sequence. We can try to contig them, but we clearly won't be able to because we were unable to generate any sequence for that region in the middle. And so, yes, we know that these particular bases are on the, in this case, left side of the sequence, and those other bases are on the right side of the sequence, and we know they kind of fit that way in their orientation, but we simply don't know what's in the middle. So we're left with this gap that we're unable to fill without another set of primers. So if you did put in another set of primers and generate that third sequence that overlapped the other two, then you could be able to fill this out into a complete contig. Okay, now that we've discussed Sanger, or chain termination sequencing, let's talk about one of the hottest new areas, which is next generation sequencing, or NGS. There are a number of different sequencing chemistries that fall under the next generation sequencing umbrella. We won't be discussing all of them. We'll only discuss the Illumina MySeq platform in detail. But all these different methods of doing next generation sequencing have a few things in common. All of these different technologies are high throughput, massively parallel methods for sequencing DNA. And each of these different methods produces between 1,000 and billions of short DNA sequences. And those short reads are all contig together later in order to form the entire genome. And so all of these different sequencing methods have a few steps in common. The genomic DNA is first extracted from your source organism. Then the DNA is usually sheared into very small pieces, and adapters are added that are specific to the chemistry for that particular platform. Then a massively parallel sequencing step occurs in which all of those different reads are sequenced. Then the sequence from each of those reads is analyzed and aligned together in order to form the complete consensus contig of the genome. As I just mentioned, we refer to the individual sequences that are generated as reads, and we can combine these to form extremely long contigs that eventually can comprise the entire genome that you're trying to investigate. So in the diagram on the right, each of these little black bars indicates a single read. And as I mentioned, for the Illumina platform, it's about 250 base pairs. So for the sake of example, let's assume that all of these are on the order of 250 base pairs. So that particular read that we're looking at there, we can take a look at that sequence and assume it's this. And part of the reason that next generation sequencing is so powerful is because of the accuracy that comes from reading the nucleotide so many times. So, in this particular example, we have a single sequence. If we also look at the read that is directly above that on the diagram, we could have this sequence, for example, and you see that the entire length of this overlap is identical, and you only have different sequence at either end where these two reads are not overlapping. But we can uh, add more reads. So if you take a look at the one above that, now you have extended to the left the region that has been read, and now you have that region that was previously read only once, now that's been read twice. And then this particular area in the middle has now been read 3 times. And in this case, all of the nucleotides in this 3 overlap region are identical. So, for example, if we look here at this particular nucleotide, we have 3 Gs. So, the fact that this particular nucleotide out of the genome was read 3 independent times and came up a G each time gives us pretty high confidence that that particular nucleotide is in fact a G. Now, with next generation sequencing, sometimes you do get, for whatever reason, a nucleotide that is incorrect, but that is usually drowned out by the vast number of correct letters. So, for example, here, if we added one more nucleotide, it could be an A rather than a G, and the software basically makes a decision and says, do I trust the 3 Gs or the 1A? And for the sake of example, that's not terribly confident, but more often than not, it would be several hundred Gs and 2 or 3 As, and so the software has a much easier way of making that decision. So in this particular case of the example, we've read that G 3 times, so we're pretty confident in that. But if you look back at the diagram, there are areas of much, much higher coverage. So if we wanted to read a nucleotide, for example, in this location where the arrow is pointing, we take a look at our coverage at that particular area, and it's extreme. So whatever nucleotide comes up as the consensus in that location is very, very confident a call. And so oftentimes this coverage can be between 200 to 400 times. I've seen coverages as much as 1,000 to 10,000 times, at which point you're essentially as certain as you can be that the nucleotide that is being reported at that location is in fact at that location. You see here a picture of the Illumina MySeq platform. As I mentioned, it generates millions of short reads on the order of 250 base pairs each. You see on the diagram at the bottom, conceptually, the reads aligned up against a reference genome sequence for easier processing. As you can imagine, generating this vast amount of data in a few hours does require some high performance computing capacity, but doing that does allow sequencing of massive numbers of DNA sequences, including entire genomes, at a fraction of the cost that Sanger sequencing would allow. Here you see the flow cell used by the MySeq platform. It's a microfluidic chip with 8 channels that contain a lawn of paired oligonucleotide primers, and you can see that in the diagram below. As I mentioned in the first slide of the section, the first step that occurs is obviously the extraction of the genomic DNA that you wish to sequence. After that, the adapters necessary for the reaction are added to the genomic DNA molecules. It's then run through a brief PCR, at which point it's then loaded onto the flow cell. Those adapters that were bound to the genomic DNA are complementary to the oligonucleotide primers that form the lawn within the flow cell channels. So this allows the genomic DNA to bind to the flow cell lawn. The individual DNA molecules will bind to the oligonucleotides within the lawn in a fairly random pattern. As you can see, not all the spots within the lawn have been taken up with DNA strands. So you have these particular strands in isolation, and then the strand will fold over and bind to one of the adjacent nucleotides on the chip. By doing that, the DNA molecule is now bound to the flow cell on either side of its sequence. Once it is folded over like that and bound to one of the adjacent oligonucleotides, the DNA strand is then filled in via replication, so it becomes double-stranded rather than single-stranded, and this forms what is called a double-stranded bridge. After that, this particular double-stranded bridge is denatured, so now there are 2 copies of the same DNA. Of course, the sequence isn't exactly the same, they are complementary, but if you recall, one of the points I made about a DNA sequence is that it inherently contains the instructions to make a copy of the other strand, and so for the purposes here, we can essentially consider these, even though they're complementary, to be the same DNA sequence. And the reason for that is when these fold over and bind to one of the adjacent oligonucleotide primers on the chip, then the replication will fill in the opposite strand, and then you'll end up with 2 double-stranded bridges which are identical to each other. This occurs multiple times repeatedly, so you have those strands will denature, bind to one of the adjacent oligonucleotides, fill in via replication, those will denature again, and then you can imagine here on this slide, this entire region here on the chip forming through multiple of these cycles an area that has the same sequence repeated a whole bunch of times. We refer to that area of essentially the same sequence as a sequence cluster. So once that cluster has been generated, the final denaturation step occurs so that all the DNA molecules are single-stranded. Then all the reverse strands are cleaved and washed away, leaving a cluster comprised of only the forward strands. So now you have a single sequence for easier sequencing. After that, a sequencing primer is added to the known sequence of the adapter at the non-bound end of the molecule, and the sequencing replication process can begin. This particular slide is sort of like Sanger sequencing in that the DNA sequence is read a single nucleotide at a time, obviously very rapidly and in parallel fashion, but each DNA sequence is read a single nucleotide at a time. So you have a fluorescently labeled DNTP that adds to the extending strand. It is then read, and the fluorescent dye will then be cleaved off, and the new nucleotides can come and be added. So it's not like chain termination sequencing. It's more like chain pause sequencing, in that the extension will be paused while the nucleotide is read, and then the dye is removed, and then the next nucleotide will come and extend the chain. At that point, it will again pause, read the label of the nucleotide in the new position, and then cleave off the fluorescent dye once again, at which point the cycle will continue multiple times until the entire sequence has been read. So now let's take a brief look at an even newer DNA sequencing technology called single molecule sequencing. The way that single molecule sequencing is typically performed is sort of the opposite of the other methods we've discussed, in that it's not based on the extension and synthesis of a DNA strand. Instead, it's based on the enzymatic breakdown of a DNA strand. So each of the nucleotides are cleaved off of the template DNA strand, and when they're cleaved off, they emit a certain fluorescent signal, which is then read and allows the identification of which nucleotide was cleaved off of the DNA strand. So a single molecule of template DNA, hence the term single molecule sequencing, is bound to a bead, which is then held in place by a laser called a trap laser. Then exonuclease is added to the system, and the DNA is degraded one nucleotide at a time. The nucleotides released are transported via electroosmotic flow to another laser. When the released nucleotide passes through that excitation laser, it emits photons that correlate with the specific nucleotide that is being excited. That emission is then read and interpreted as the specific nucleotide. Through the repetition of these steps multiple times, an entire DNA sequence can be sequenced from a single molecule. A different form of single molecule sequencing is single nucleotide sequencing by synthesis, which is sort of a hybrid between the Sanger sequencing that we talked about earlier and the single molecule sequencing method that I just described. As you can imagine from the name, the process occurs by synthesis, not by the breakdown of the DNA strand, as we just talked about. So in this method, the fluorescent nucleotide is read as it is incorporated into the growing strand. So the template strand is also attached to a bead, and then the synthesis of the complementary strand, complementary to the template strand, is performed, and the fluorescence from the nucleotide being attached is read. Okay, so here are the references for this lecture, and I hope you found it useful, and let me know if you have any questions on gel electrophoresis and DNA sequencing. Thank you.